Hello everyone. My name is Shubhangi Kesarwani. I'm from Prayagraj, India. And today we are going to study about a very interesting topic as you can see it on your screen, that is DNA and RNA structure. Okay? So before we start, look at your hand for 5 seconds and now tell me, why doesn't anyone else in the world have hands exactly like yours? Because everyone is different, right? And it might be your genes or due to your parents' genes, right? Very good. Now, my next question is, you and your parents look similar, right? But not identical, right? So, where is the instruction book that tells your body how to cook? Okay. So what happens is, inside every cell of our body, there is something which is called as biological Google Drive. You know what is Google Drive, right? So there is biological Google Drive which is present in our body that actually stores the information. What kind of information? That instruction is called as DNA. Okay, so what is DNA? We used to see a lot of things about DNA. So, what exactly is DNA? DNA is like a recipe book of your body. What does it mean? Means it will contain some of the instructions for your eye color or your hair type, right? Even your height as in, you must have seen some people are tall, some people are short, and some people are of medium height, right? So that's how your cell works. Now tell me, if DNA is the recipe book, okay, so where do you think it is kept? Any idea? In your kitchen, or your pocket, or in your blood? Yes, so DNA is present in our cells. Okay, DNA is present in our cells, or it could be present in our nucleus. I'm assuming that you know what is nucleus, right? So it is present inside our nucleus, which is acting as the control room of the cell. All right. So now we will discuss about the DNA structure. How does it look like, and how it has discovered? Okay, how are we studying this? Okay, so scientists didn't just guess the DNA shape, that it must be looking like this. No, it doesn't just happen overnight. And they did just guess the DNA shape. So there is one scientist which is called as Rosalind Franklin, as you can see it on your screen as well. So what happens is, this scientist took an X-ray of X-ray picture of the DNA. X-ray picture of the DNA. So tell me one thing, if something gives you a repeating pattern in X-ray, do you think it could be messy, or it is well organized? Yes, it will be well organized, because we are saying that it is repeating and it is regular. Okay, so if something is continuously repeating and it is regular, it means that it is going to be organized. Exactly, that's how we learned that DNA has a regular spiral shape. Do you see? This is a kind of regular spiral shape. Yes, and this phenomena, Rosalind Franklin gave us this um, mechanism, which is called as X-ray crystallography. Okay, that's how we got to know that DNA looks like spiral shape. You know what is a spiral shape? It was like this. Hmm? That's how spiral looks like. Understood? Later on, Another scientist also came along with Franklin. His name is Edwin Chargaff. Okay. So he studied the DNA of many organisms. Okay. And analyzed the DNA samples from different species. And he found something very shocking. What was that? What was the shocking part? So he got to know that the amount of A is equal to the amount of T, and amount of C is also equal to amount of G. What does it mean? A, T, G, C. So A means adenine, means thymine, C means cytosine, and G means guanine. So why do you think that happened? Is this a coincidence? Or maybe they pair, right? Just like our dance partner. Okay. So the amount of T is always equal to the amount of A, and C is equal to G. This is how the structure looks like. Okay. So let's say DNA is a kind of long train. Okay. DNA is kind of long train. Okay. So why do you think what do you think each of the unit, or generally we call it as coach, right? What it is called? We call it as nucleotide. Okay, so each nucleotide, very important. If somebody asks you that what is nucleotide, so nucleotide basically made up of sugar, sugar, phosphate, and base. Just now we have discussed about this ATGC. These are nothing but these are the bases. Okay, these are called as bases. All right. So if we say that DNA is made up of nucleotide, and nucleotide is made up of all these three structure, which is sugar, phosphate, and base. What are bases? Here A, T, G, and C. Okay. Just now we have discussed. See all these four structure A, T, G, C. All right. Okay. So bases are the letters, as you can see. A, T, G, C. All right. So as you can see, nucleotide is made up of purine and pyrimidines. In the structure, you can see the difference. Okay, in purine, there is double ring like structure. Double ring as in like this. But in pyrimidine, it is like just single ring, right? Purines are kind of a bigger structure, double ring structure, and which include purine will include AG, means adenine and guanine, and pyrimidine will include cytosine, thymine. And what is this U? I will discuss this in upcoming minute. Okay, so the U means uracil. U means uracil. All right, this we will discuss in few seconds. Okay, so pyrimidine include cytosine, uracil, and thymine, and purine includes A and G, means adenine and guanine. Absolutely right. Okay. So now we have come to the nucleotide pairing. It says the base pairs are held together by hydrogen bonds. So tell me one thing, why don't A pair with C and G with T? because the shapes must match like puzzle pieces. All right. So generally what we know that A when A combined with T and G combined with C. This part we already know, right? So the thing is, in nucleotide pairing, what we study about the hydrogen bonding, how these purine and pyrimidine are joined together. As you can see, A always binds with, make a bond with T with double bond, with double hydrogen bond. Okay, and G always make a bond with cytosine with three hydrogen bond. Okay, see this image, G and C are joined together with one, two, three hydrogen bond, right? And A and T are also joined, but as you can see, one, two, only two hydrogen bond, right? So this is the thing. So tell me one thing, out of these two, A, T and G, C, which one do you think is stronger? G, C or A or T? So the answer is G and C. Absolutely right. Why G and C? If something has a stronger bond, it means that they are going to be held together in a stronger way. Okay. And this one is having double bond, and this G and C having three hydrogen bonds. Okay. So G and C is going to have a stronger hold, right? Absolutely correct. I hope you have understood till here. Okay. So now there is one thing related to the discovery of the DNA structure, which is, as you can see, Watson and Crick combined the findings of Franklin. Okay, this part we have studied already about the helix shape. Franklin told us how it looks like. He told us it looks like a helix shape. Okay, and Chargaff told us about the base pairing. Okay, to create the first 3D double helix model of DNA. So tell me, does this DNA looks straight? No, absolutely not. It is kind of what structure? Spiral. Okay, firstly, tell me if this is a ladder. This is the normal ladder we know how it looks like. Absolutely right. Okay, so just imagine there is a ladder and you are just spiraling it. You are just, twisting it. So it will look exactly like this. Stair is converted into the helix shape. So how does it look like? A spiral staircase with having a twisted ladder. Okay, just like a phone cord. In earlier days, we used to see the phone cord, right? So which one? Which helps you imagine better? Okay, so what happens is Watson and Craig gave us the 3D model. Okay, it gave us the 3D model using what ideas? Franklin and Charga data. Okay, that's how he got to know about these things. Okay, so what is hydrogen bonding? If DNA strands were stuck somewhere very tightly, could they just open during copying? No. So hydrogen bond have are weak. Okay, they are not that strong. We are talking about hydrogen bonds here. So hydrogen bonds are weak, they are easy to break, so that DNA can unzip during the process of replication. So the replication process, we will discuss this in the upcoming classes. Okay, so just now we know how little bit just replication. So replication is like you are making a clone of something. Okay. So how you are making a Xerox copy of something, right? So that is the replication process. So here we are, what happens is DNA is making a clone or the Xerox copy of itself. Okay. So if the bonds are, just imagine if the bonds are very strong, then how the structure is going to unzip and how they are going to make the copy of themselves. So the hydrogen bonds have to be weak. Okay. So what is the function of DNA? Till now we are discussing about what is DNA and also what is the function of DNA. So DNA main job is to store the information, okay, and pass it to the next generation. We generally say that you have got this um, feature from your father or your mother. Okay, that's how you are passing the information from one generation to the next generation with the help of DNA. All right. So if DNA didn't copy correctly, what might happen? If the DNA didn't copy correctly, what might happen? What are the possible things? We can get mutation, right? Mutation. What else we can get? We get different type of diseases, right? What else we can get? Changes in the traits. What are traits? Traits means general characters. Changes in traits. All right. So these things would happen if they are not um, copied correctly. Okay, these could happen. Okay, and there is one exception, as you can see it on the screen. RNA is the primary source of heritable information in some of the virus. Okay, in the previous page, we have discussed about the pyrimidine, right? CUT. Okay, so DNA, we have DNA. See this very carefully. We have DNA and we have RNA. This is very important, okay? So DNA has A, T, G, C, all these four bases. But RNA has A, G, C, but something is missing here, right? So in place of T, in place of T, we have U in RNA, means uracil. So T is replaced by uracil here. Okay, this is very, very, very important. And this is a very main difference of DNA and RNA. Okay, very important. Okay, so same thing, it says RNA is the primary source of heritable information in some of the viruses. See, 90% of the viruses have RNA as the um, RNA as the heritable information, okay, which passes from one generation to another. But us, which one will be the primary source of heritable information for humans? It will be DNA. But for viruses, mostly it is RNA. Okay, that is the thing they are talking about. This is the exception. Generally, DNA is the primary source, but in but in viruses, RNA is the primary source. So this is the thing. I hope you get it. Okay, now come to the prokaryotic versus eukaryotic DNA. So basically your cells, which is eukaryotic cells, keep the DNA safely inside the nucleus. Okay. And what about bacteria? Bacteria. Bacteria are considered as prokaryotic. Okay. So it will be having no nucleus. DNA just floats in the cell. Okay. So which one sounds more organized? Eukaryotic or prokaryotic? Tell me. So eukaryotic is much more organized because the DNA is found in the nucleus. This is the basic thing. DNA, we are eukaryotic cells, right? And pro means they are primitive cells. We are not primitive, but if you take the example of what's that called? Yes, the prokaryotic cells, which is bacteria. Okay, so they are very primitive. They are not as advanced as us, right? So that is why we are calling them as primitive, and we are advanced organism, right? So DNA, the DNA of our uh, the DNA is present inside the nucleus of our body. Okay, so they and how does it look like? The chromosomes will be linear. Okay, and here the chromosomes are circular. They will be in circular shape, but here the DNA will be in linear shape. That is the main difference. Okay, this is very, very, very important. So there are some prokaryotics. Okay, and there are some of the eukaryotic that also contain plasmids. Okay, so what are plasmids? But this is very important. These are small circular DNA molecule that separates from the chromosome. So the thing is, basically the plasmids are found in plasmids are generally found in bacterias. Okay, generally found in bacterias. Okay, so bacteria also have this extra mini DNA circle which is called as plasmid. Okay, so imagine you have a USB with extra skills, right? So plasmid is going to carry that antibiotic resistance. Okay, so that's how bacteria become super bugs. So as you can see it on the screen, plasmid replicates independently. See, this is the chromosomal DNA, okay, which is in circular form. Just now we got to know. Just now we got to know in the previous that the chromosomes are in circular form, but in eukaryotes, which is we are eukaryotes, okay, all the mammals and all. So we are eukaryotes and our chromosomes is in linear form, not in a circular form. Okay, always remember that. Okay, so this is the chromosomal DNA, but here the plasmids are kind of in circular form. Okay, so these are primarily found in the prokaryotes, so it will contain some of the genes that may be useful to the prokaryote, and when it is in particular environment, but maybe they are not, but they but may not be required for the survival. Okay, so this is how the plasmid works. Okay, we can also manipulate plasmids in the laboratories. Okay, so plasmids can be removed out from the bacteria, and the gene of interest, whatever we trying to make, like whatever type of gene we want to make, we can insert in that plasmid, and that phenomena is called as recombinant plasmid DNA. Okay, so this is how plasmid can also carry the antibiotic resistance. Just now I have told you that's how we can make bacteria some of the superbugs. Okay, that's all about this topic, and after that I'll give you one more video of upcoming topics which is related to the DNA and RNA, more about this topic. Okay, so thank you so much for watching this video. I hope you understood. Thank you so much.